Reverse transcription or teminism. Reverse transcription or teminism is a unique phenomenon of gene expression found in a group of RNA viruses called retroviruses. These viruses violate the central dogma during gene expression. Human immunodeficiency virus (HIV) and tumor viruses such as mammary tumor virus (TMV), Rous sarcoma virus (RSV), avian retrovirus discovered by Rous found in chickens, etc. are retroviruses. A retrovirus contains two identical RNA molecules linked in a complex. In retroviruses, gene expression is a reversed central dogma in the initial phase. According to the central dogma, information flows from DNA to mRNA to protein during gene expression, but in retroviruses, there occurs a reverse flow of mRNA to DNA. This is called reverse transcription, a term invented by Howard Temin. Based on the studies of RSV, Temin in 1963 postulated that the growth cycle of retrovirus involves an alternation between RNA and DNA generations. The DNA called provirus is only an intermediate molecule and the DNA can be synthesized using RNA template. This hypothesis, often called Teminism, was later on confirmed when Temin and Baltimore in 1970 independently discovered the enzyme reverse transcriptase in several avian and mouse retroviruses. This enzyme can catalyze RNA dependent DNA synthesis. Retroviral transcriptase are peculiar in that all of them have considerable similarity in amino acid sequence. In reverse transcription, genomic viral RNA acts as a template and guides synthesis of DNA strand called complementary DNA or cDNA. This this RNA dependent DNA synthesis may be called reverse transcription. It is modified by the enzyme reverse transcriptase, which functions as an RNA dependent DNA polymerase. The cDNA strand initially remains complexed with the template RNA, forming a DNA RNA hybrid. Reverse transcriptase now functions as a ribonuclease and removes the RNA from the complex. The cDNA then serves as a template and guides the synthesis of a complementary companion strand to form a linear DNA duplex called DNA provirus. At this stage, reverse transcriptase functions as a DNA dependent DNA polymerase. Reverse transcriptase is capable of initiating DNA polymerization. So, its polymerase activity requires a primer onto which DNA nucleotides can be incorporated. The commonest native primer is a tRNA fragment. The linear DNA duplex is only an intermediate molecule. It soon circularizes and makes its way from a cytoplasm to the nucleus of the host cell. Soon it gets integrated with the chromosomal DNA of the host cell with the help of enzyme integrase. Integration is a normal part of the life cycle of viruses necessary for transcription. The gene order in the integrated DNA is the same as the gene order in the viral tRNA. The integrated proviral DNA undergoes transcription with the help of host cell machinery, forming viral mRNAs and viral genomic RNAs. Viral mRNAs undergo translation producing viral proteins. This brings about viral gene expression. Two copies each of viral genomic RNAs get packaged with viral proteins to form retroviral particles. This in turn causes the transmission of the virus to new host cells. Retroviruses are called plus strand viruses since their genomic RNA codes for protein products. The single stranded cDNA synthesized on viral RNA is called minus strand DNA. The second DNA strand synthesized in it is called plus strand DNA. So, retroviral genomic RNA undergo reverse transcription to form cDNA. Later, that duplication will be happening. Then, DNA provirus integration with host cell DNA, transcription. Later, viral genomic RNA is formed. Viral mRNA is formed which undergo translation to create protein. But that viral genome RNA packaged with the protein once again form retroviral genomic RNA. So this is a quick recap about reverse transcription process or feminism. So if you paid attention, you learned about this process. what is the peculiarity of this process. In some what sort of viruses this is happening. So thank you for listening.